Hello and welcome to Year 11 Biology, the Mind Map of Cells. Take a picture now if you want to, otherwise we're zooming in. Okay, we're going to come up here to the history of cells. Robert Hooke was the first one to see a cell in a cork plant. Then Van Leeuwenhoek was the first one to see a single-celled organism, and he called it an animalcule. Then Brown saw the first nucleus in a plant cell. Schwann and Schleiden then developed cell theory for the most part, which was then added to by Virchow, who added to cell theory about cell division, in that all cells come from pre-existing cells. Then Rusker and Knoll developed the electron microscope that allowed us to see organelles in minute detail, and then Margulis first postulated the endosymbiotic theory. Now, as we move on to cell theory itself, we can see that all life is composed of cells. All cells have functions, and all cells come from pre-existing cells. Now, a cell's life, for the most part, is what we call interphase. It is 80% of a cell's life. It contains G1, which is the growth phase 1, S, which is the synthesis of DNA, when DNA goes from being single-stranded to double-stranded, to growth phase 2, which is the second stage of growth. Now, what characteristics do cells need to survive? All cells need water. In fact, 65 to 75% of a cell is composed of water, and it's because it's a solvent for all of the molecules inside cells. All cells need ions to make molecules. All cells need a metabolism to make energy. All cells need to maintain a stable cytoplasm for homeostasis. All cells need genetic material, that is DNA. All cells use cell division, which is part of cell theory. All cells have organelles, which goes to the theory that all cells have functions. All cells respond to their environment, and all cells must remove wastes, and these wastes are normally nitrogenous. Okay, then we're getting down to the different types of cells. First of all, we have bacteria and bacteria-like organisms called archaea. They are unicellular, which means they are made up of one cell. Bacteria have a cell wall of peptidoglycan. Archaea have a cell wall of uncommon lipids. They both have ribosomes, and they both possibly have hairs, and there are 3 types of hairs that we'll talk about later. They both have genetic material, and they have that in the form of a nucleoid, which is a large, massive DNA attached to the inside of the cell membrane. And they also have plasmids, which are circular pieces of DNA that are self-replicating. Both prokaryotes and archaea can be autotrophic or heterotrophic. Autotrophic means they make their own food. Heterotrophic means they eat other cells for food. Okay, then we move down to the kingdom of eukaryotes. Eukaryotes have membrane-bound organelles and a true nucleus. First, we have protists, which are the junk drawer of classification. If anything doesn't fit in another classification, it gets put into protists. They are single-celled and aquatic. Then we get down to fungi, which have a cell wall of chitin. Fungi can be heterotrophic, which means to eat other cells, or they can be saprophytic, which means they decompose dead matter. Then we get on to the 2 main ones we know of, which are animals and plants. Animals have no cell wall, but they do have lysosomes. They do have centrioles. They do have a small transient vacuole. They are heterotrophic and they are motile, which means they move. Plant cells, on the other hand, have a cell wall made of cellulose. They have chloroplasts for photosynthesis. They have a large central vacuole. They contain plastids, which are like vacuoles, but they contain pigment as well. And they are photosynthetic due to the fact they have chloroplasts. All right, over here we've got diagrams of the different organisms you need to know. We've got a bacterial cell. We've got a protist, a generalized protist. We've got fungi. We've got animal cells. And then we've got plant cells. Okay, we're getting on to the different types of organelles now. Right, so first of all, we have a nucleus that controls the cell using DNA to make proteins. We call that gene expression. Inside the nucleus, there is the nucleolus that makes rRNA and therefore makes ribosomes. Some cells have a cell wall. Fungi has a cell wall made of chitin. Plant cells have a cell wall made of cellulose. They all have a cell membrane composed of a phospholipid bilayer that uses the fluid mosaic model. They all have ribosomes that have no membrane, and that's why they are found in all cells, including bacterial cells, and ribosomes make proteins. Now, here are the hairs we talked about earlier. There are 3 different types that a cell can have. They can have fimbriae, that is used for attachment. They can have pili, which are hairs for transformation. Transformation is the swapping of genetic material, which is plasmids. And they can have a flagellum, which is used for movement. Eukaryotic cells have an endoplasmic reticulum. They are transport channels that are linked to both the nucleus at one end and a Golgi apparatus at the other. There are 2 types of endoplasmic reticula. There is smooth that has no ribosomes, and they make lipids and carbohydrates. Then there is the rough endoplasmic reticulum that has ribosomes encrusted on the outside, and that makes and transports proteins. Next, we have a mitochondrion, which is endosymbiotic. It is the site of ATP synthesis and contains ATPase, which is an enzyme that makes ATP, and that enzyme is found on the cristae, which is the folded internal membrane of the mitochondrion. There's the folded internal membrane. Then we have chloroplasts, which are also endosymbiotic. They are the site of photosynthesis and contain chlorophyll embedded in the thylakoid, which is the folded membrane on the inside of a chloroplast. Here we have the thylakoids. Animal cells have lysosomes that break down food and broken organelles. Peroxisomes destroy the dangerous molecule H2O2, which is hydrogen peroxide, that would normally kill a cell. Centrioles are found in animal cells, and they make spindle fibers for cell division. Plant cells have something called centrosomes that also do a similar function. As we said before, both plant cells and animal cells have vacuoles. Plant cells have a large permanent central vacuole that contains mainly water, and the vacuole in animal cells is small, transient, and peripheral. Then we get on to plastids, which are like vacuoles but also contain pigments. Golgi apparatus, or sometimes called the Golgi complex, is linked to the endoplasmic reticulum. It can store, modify, and package organic molecules for transport, and they are transported in vesicles. Here are some vesicles here. Then all cells have to have a cytoskeleton, which is made up of tubules, which provide structure for the cell. Right now we get on to microscopes. Parts of the microscope, we've got an eyepiece lens, otherwise called an ocular lens, that is for the most part times 10 magnification. We have a nose piece that holds objective lenses, and there are 3 types of objective lenses, being times 4, times 10, and times 40. Then we have a stage that has clips on it to hold the slide in place. We have a light source. Today, modern microscopes have a light as the light source rather than a mirror, and that shines light onto the slide so we can see it. You'll see on the side there we have focus knobs, being the coarse and fine adjustment to increase the sharpness of images. And we also have an arm that is the backbone that holds the other parts of the microscope in place. All right, the first thing you need to do when you get a microscope is to work out the field of view. The field of view is the distance from one side of the image to the other. You can calculate the distance from one side to the other using a mini grid. Here is a diagram of a mini grid on a microscope. The field of view is made up of big squares and small squares. The big squares are 1 millimeter by 1 millimeter, and the small squares are made up of 0.1 millimeter by 0.1 millimeter. And a micrometer is 1,000th of a millimeter. Next, we have total magnification. The total magnification is the eyepiece lens times the objective lens, and we can end up with times 40, times 100, or times 400. And as a general rule, as the magnification increases, the field of view decreases. So if I change the magnification by a factor of 10 from times 40 to times 400, the field of view will go down by a factor of 10 from 4,200 micrometers to 420 micrometers. We also need to know how to make a wet mount. Now, to make a wet mount, we obtain a specimen, and it should be thin, and place it on the slide. Then place a drop of water on the specimen. Then place the cover slip at 45 degrees over the sample, we can see that here, and then slowly lower the cover slip down with forceps. And make sure that there are no air bubbles because they will ruin your image. The last thing you need to know how to do is stain. Once you've got the cover slip down on your specimen, place a drop of stain on one side near, actually touching the cover slip. Then place a piece of paper towel on the opposite edge and that will draw through or wick through the stain. Leave the stain on the specimen for 5 minutes before you try to look at it so that everything is evenly stained. Down the bottom here we have the tree of life that explains how everything relates to each other. And as we move out, here is the mind map. Thank you for listening and I'll see you next time.